now. This talk will be filmed, as I understand, so if any one of you has any objections or don't want to be filmed, maybe you can move in the back or something. So I'm happy to introduce Pete Gagnon. You are at BIA Separations and you're the Chief Scientific Officer there. So here we will hear more about the ways that host cell contaminants interfere with purification and product quality of EVs. Most welcome.
How does this work? Thank you very much. Thank you for the invitation. I'm going to give you a really different sort of a perspective on exosomes. I'm going to cover a lot of stuff. I'm going to go really fast. But don't worry, because you can all have the slides, and I'll be around the next couple of days if there's questions that go beyond the scope of what we can cover right now. From an industrial perspective, exosomes represent one of the toughest challenges in town. They overlap, as you've heard this morning, in size and chemistry and density with most of their major contaminant classes. Also with lipid envelope viruses, chromatin aggregates, all kinds of stuff. In addition to that, they don't have uniform surface chemistry. Some of them have phosphatidylserine on the surface, some don't. That affects their surface charge. Besides that, they're all the specific protein expression on the surface. And by the way, I agree with you about exosomes being team players, so when we get to that. Also, they don't tolerate chemical extremes very well, so that restricts the range of purification tools. And most of the purification tools that have been designed over the decades for industrial purification of genetically engineered materials, they're all built for proteins. And so exosomes are anywhere from like 15 to 50 times bigger. And that really restricts their binding capacity, creates shear forces and things like that. So what we need are new options there. In addition to that, process monitoring is another challenge. It's really hard to get like nice black and white assay results from exosomes. They absorb very little UV light. That makes them difficult to detect with conventional chromatography. On SDS page, they produce lots of bands, so you can't, again, monitor them very easily. Western blotting helps, but you still have a diversity of types there. And particle detection methods, they can discriminate among vesicle classes, but they still require cross-corroboration to make sure you have the right vesicles in there. So today, I'm going to show you a few things. I'm going to show you some new analytical approaches that overcome these limitations. And mind you, I'm not talking so much about biology. I'm talking about limitations from the perspective of how do you purify these things and how do you monitor the purification process. So one of these assays gives you easy discrimination of vesicles from the majority of non-vesicle contaminants. Another one, exosomes from other vesicles. More that can give you quantitative size distribution, and another one that can discriminate empty from full exosomes. I'm also going to introduce some new purification tools, including one that addresses a purification issue that's really not been addressed so far as I can tell in this field. Okay, so I'm going to start off with a little discussion here with multi-angle light scattering. It's one of the methods we use to monitor chromatography, and it adds a nice dimension to it. So light scatter is proportional to vesicle size. So vesicles being bigger, they produce more light scatter than proteins, and that amplifies their signal proportionately. So this is just a pictograph of that. You get these kind of things where you can actually determine signal intensity. You can also calculate radius of gyration. And you can estimate even molecular weights if it gets to it, but mostly we don't concern ourselves with that one. So here are some profiles from mols. What you're seeing here, this is an overlay. The red profile is where we just looked at UV. And so what you see is where you have high mols but low UV in this sample. That's an indication, that's a signature of vesicles. So you'll have high mols but low UV. If you have high UV but low mols, that means you've got proteins and smaller things in there. So you can discriminate those things very easily. And then there's another aspect of this that you can look at. Oops, and okay. It will give you actual plots of the sizes of materials that are in there. So in this fraction from size exclusion chromatography, what you can see is there's a range of sizes here. Some of these bigger things, those correspond to microvesicles, apoptotic bodies and stuff like that. Then you have this material that's in the same size range as exosomes. And then out here you get bits of garbage and stuff that's much smaller than that. You'll see a lot of examples here today. Here's a multi-angle light scattering with size exclusion chromatography. The column's not anything special, it's just a TSK column. This was filtered serum-free HEC 293 harvest. And over here, this is where the Here's where the exosomes come out, but they also come out with apoptotic bodies, microvesicles, high molecular weight chromatin aggregates, virus particles, lipid vesicles, and cell debris. So if you're using size exclusion, you're getting more than exosomes. This material, all these things are in the void peak, where you elute things that are too large to go into the pores of the media and whatnot. And then in this size range here, you have chromatin aggregates and proteins, peptides, smaller stuff as you go over here. So this method can't really discriminate the size, but at least the moles helps you understand where the vesicles are. Here's an enhancement of that. What we did in this case is we injected the sample with a CD9 FITC in advance, and we monitor both the multi-angle light scattering and fluorescence at the same time here. And you can see there's a really strong signal from the CD9 that tells us this is where you have vesicles with the moles response. Those overlap very well. And that's good. CD9 basically picks up all vesicle types. Multi-angle light scattering picks up all vesicle types, so they should agree, and they generally do agree. So it makes a good experimental control. You can also use this technique of using immunofluorescent conjugates in conjunction with size exclusion chromatography to show where your contaminants reside. So in this case, what we did here is we added LDL, anti-LDL FITC to this material. And what you can see here is that, there is some signal. You've got everything that comes before where this black bar starts. That's showing you have LDL vesicles over there. And you can see there's definitely a component of them that overlaps with this vesicle fraction. That means it's contributing to the mole signal. So moles doesn't tell you what's in it, it just tells you it's big. And what this result tells us is that where we get exosomes, we also get liposome vesicles. Here's another case where here we took an antibody for apoptotic bodies. And what you see here is there's a lot of stuff that's smaller than antibodies. Apoptotic bodies are mostly a lot bigger than exosomes, but what happens by the time you get to a cell culture harvest, they're massively degraded. So most of them are quite a bit smaller than that, so you see debris all the way across here. But there's still a significant subpopulation that's out here with the exosome population, which means it's also contributing to your mole signal. Microvesicles, same thing. These are also bigger than exosomes in vivo, but they're highly degraded by the time you get it to a cell culture harvest, and still they have a subpopulation that will contribute to this mole signal. So they're in there as contaminants. Here's a case where we look for DNA. This is a little bit different use of fluorescence. So we spike the sample before putting it on the size exclusion column with picogreen. Picogreen fluoresces only when it's intercalated into DNA. And you can see that there's a positive response there that, again, is contributing to it. But you see all this DNA across here. What you're seeing is the remnants of the chromosomes from the cells that died during harvest. And what happens is they're occupying the same size range. They get up to 400 nanometers. And here you see an agarose gel here. So this first one, this is the whole culture. And you can see there's some single nucleosomes still in there, nucleosome doublets, triplets, and whatnot. And you can see there's some size fractionation, and you get RNA out here as well. So it's kind of handy to be able to know where the RNA is. Okay, so now we're going to take a look at some of the common filtration methods. A couple people have made some comments about them this morning. Tangential filtration. We looked at a bunch of membranes. The one we liked the best was this one. It's a 750,000 molecular weight cutoff. If you use smaller cutoffs, you'll retain your exosomes, but you'll retain a lot more garbage in there too. So what we see in here, if we look with moles, is like, yep, here's before TFF. We can see the vesicle fraction. You can see all these smaller contaminants. After TFF, still have the vesicle fraction. You can see the UV absorbance is a little smaller. That's good. But you still see there's a lot of protein here. And it turns out that this material is all 254 nanometer dominant. Long story short, that's DNA doing that. So what it tells you is the nucleosomal debris from the dead cells is persisting, and the reason it stays after TFF is because it sticks to the membrane. This stuff's unbelievably sticky, and I'll tell you more about that a little bit later on. Here's where we took a commercial kit using a low pH precipitation step. It uses some other stuff too, I think. But what you see here, this is the same starting material that we saw before. So here's your vesicle fraction. Here's what it looks like afterwards. And you see a lot of chromatin here. The reason you see a lot of chromatin is because chromatin also precipitates at low pH. So this method is good for getting rid of proteins and stuff like that, but it leaves you with the worst contaminants in the system. And you also see that, this actual UV peak here, that's less than 10% of the total UV absorbance. So that's not a good indication. It tells you you're not getting a lot of purification here. Here is another commercial purification system that uses polyethylene glycol. A lot of you know what this is. And you see a similar case. It's a lot better. So polyethylene glycol does a really good job getting rid of a lot of small protein contaminants or whatnot, but it also co-concentrates chromatin, and it also co-concentrates all your other vesicle types with this. So they'll reduce your contaminant load, but if you're looking for specific exosome materials, neither one of these systems works very well for that. So what is chromatin? You see I'm emphasizing chromatin as we go along. What is chromatin and why should you care? So chromatin in cell culture harvest is the remnant of the chromosomal mass from the host cells. And its basic subunit is a nucleosome. You can see here, this is a nucleosome diplex where you have two of these. So you have a histone octamer, that's got about 1.6 wraps of DNA around it. There's these interlinker sequences that are 20 to 80 base pairs. And this is what it looks like in cell culture media. So like I said, you have some single nucleosomes. This is all your chromosomal debris. You have some that are two nucleosome arrays, which would be like what's illustrated here, 3s, 4s, 5s, and on up from there. This is important because DNA is really super electronegative. The minor groove is hydrophobic. DNA also participates in hydrogen bonding, metal affinity, van der Waals interaction. Chemically, it's very sticky. Histones are even worse. They're electropositive and they're extremely hydrophobic. They also participate in all these things. So what this does is it means that chromatin is like a super multimodal affinity ligand that sticks to everything. So it sticks to filtration media, chromatography media, probably sticks to exosomes to some degree, and every other surface they contact. So chromatin's a problem from that perspective. And by the way, if you're working with living systems, not so much. So you folks that are doing research with natural samples, they're in living systems. You have phagocytes that take care of dead cells and run them off to the liver to get shredded. But in cell culture, there are no phagocytes. So all the chromatin that's left over from the dead cells, it gets chewed up, it gets degraded, but there's a lot of it still there. Okay, it's a problem for three major reasons. One, chromatin protects DNA. The association of the histones with the DNA is so strong that it prevents access by nuclease enzymes. So you can treat these material with benzonase or whatever enzyme that you want, but you won't get rid of all the DNA, and the reason is because their strong association with protein protects them. Chromatin interferes with all purification methods. In the case of some affinity methods, for example, it will reduce your capacity up to 75%. It'll inflate your host cell protein concentration by 100-fold, DNA by 10,000-fold, literally. This comes from some published research, mostly with antibodies. And the worst thing of all is, like, we look at exosomes as being, like native materials. They should not be antigenic. And yet extracellular chromatin is. So if you don't get rid of this contaminant, it may cause you some headaches down the line. So we developed an enzyme treatment. It's a special enzyme and a special process to deal with this. And this shows you, again, here's the same filtered harvest, and here's the DNA numbers by picogreen. You can see the chromatin and everything like that. So after TFF, we add this material, and that drops the material down quite a bit, about 100-fold and whatnot. And you can see that because of the TFF, we got rid of most of the proteins here. But now we've treated with this enzyme. You see the chromatin's mostly gone. You don't have that there. But what you now have is a real high signal over here in the small molecule part of this profile. And what that corresponds to is nucleotides and histone proteins that have been liberated by this enzyme treatment. Then you go to a second TFF step, and you can see it clears that stuff out and a lot of the other junk, and it leaves you in a lot better place to go into a real purification step. Okay, this will give you an idea about how that looks from another perspective. So this is flow cytometry results where we looked at material. This is before the kryptonase treatment, so it's after the first TFF step, but before the kryptonase. And here's your exosome fraction down here. These are the small EVs. And what you see is that after the second one, so after we treat it with the enzyme, we've done the second one, we have really good conservation of these. The cell counts here are really quite close together. So we've seen quite a lot of indications that the treatment does not damage the exosomes. There's this paper by Jurgens et al., and you can follow up on that. And also, we have some collaborators who provided this information for us. But this will give you an idea. The point of this slide is this treatment does not damage the exosomes. Choice of chromatography media is also important. Now, we use materials that are called monoliths. Monoliths are different from porous particles. One, because flow is laminar through monoliths. They have a different architecture than porous particle materials. So they don't generate the shear forces that other materials do. Also, the channels are very large, and you're using different kind of mass transport in there. So you can get high capacity, good separation, and high flow rates. You can refer back to this. We can talk about that more. And like I said, the capacity is very good on these materials. And when I say good, I mean for exosomes, you should be looking at like maybe 10 to the 13th to 10 to the 15th particles per mil, something like that. So now, what you're seeing here, this is a chromatography separation step. It's on a positively charged medium. So this diagonal line, that's a salt gradient. And you can see that this is filtered harvest, so it's got all the garbage in it. And the mole's response is in gray in the background here. So you can see there's a lot of protein in there, but you see a lot of 254 dominance, which means there's a huge amount of chromatin still in there. So this material actually does capture the chromatin, and it co-purifies it with stuff. And in the back, you can see there's vesicles all the way across this. So that's maybe good, maybe bad, but I'll show you it's actually bad very soon here. This is what it looks like if you do this after kryptonase treatment. This is exactly the same material, same chromatography material, same separation conditions, but we got rid of all the chromatin before that and all the small proteins. So you see a really different situation here. So we're using immunofluorescence with CD63, also moles, and you see those two curves agree very well, and they tell you that the exosomes elute in the early part of the gradient with this material. Now you still see a mole's response out here, and that tells you that this is other kinds of vesicles. And we know from experience also that we've tested this material, so microvesicles, apoptotic bodies and whatnot, they tend to come off much later. The exosomes tend to come off early on. There's also an important caution in this data, and if you look at this, you'll see there's still a peak here in the front. There's some chromatin that's still left in there, but there's also a lot of protein in there. So what this is telling you is that two good vesicle detection methods, that being multi-angle light scattering and the immunofluorescence, they're both telling you where the exosomes are, and yet you've got a protein spike or another spike over here that does not correspond with those. And that's a warning that UV is not a good method for monitoring exosomes. They don't absorb much, so if you've got a lot of UV absorbance, it's probably not coming from exosomes. So if we go back now and compare the results with these other purification methods, you can see these are the ones that you saw before, the first one and the second one. And what we do is this gets rid, by getting rid of the chromatin up front, this really cleans the system up and you have a much higher quality fraction. You can also see that, the amount of this material, the sensitivities are a little bit different here. This has lower UV absorbance in it because we've gotten rid of the UV absorbing materials. Here's just a standard Western blot. We did this, so there's a moles profile where we determined the size range of this peak, and Western blots to show that, yeah, all the right exosome markers are in there. This is a really interesting one. Here we took all the immunofluorescence profiles and overlaid them for the raw material. So, all these vessels, all these things we looked at before, microvesicles, liposomes, all this kind of stuff, they're all here. You can see them in the raw material. But after purification, basically the non-exosomal vesicle material is just that tiny little bump on the baseline there. And that tells you that you have a real highly enriched exosome population at the end of this process. And all this other stuff in here, this is the antibody that has the FTSE attached to it and bits of debris and stuff like that. But you see, we do a good job getting rid of the extracellular vesicles there. Here's another case where you can use fluorescence. So these folks provided to us exosomes that had GFP engineered into them. So you can also monitor that fluorescence. That's easy to do. And it's more specific because, all of these exosome markers, they also occur on other kinds of vesicles. So if you've got an internal vesicle, it's nice. It's not as sensitive, but it can give you a different piece of information. Here's another thing we did just for fun. It's like Exosomes, you should be able to penetrate the membranes with things like ribogreen or whatnot, and you do. So you can actually detect the RNA that's inside these vesicles, and it makes it, gives it a lot more sensitivity for monitoring purposes. So here's where we took some of the beta purified exosomes, popped it with ribogreen before we put it on size exclusion chromatography, and indeed you can see there's a lot of RNA signal associated with this population here. Meanwhile, most of the host cell protein or RNA is removed by this process. Here's a case where we use bioanalyzer profiles to characterize the size distribution of this stuff. So the red profile is the filtered harvest before treatment. The blue profile there is from the stuff after the beta purification. And you can see there's a very definite enrichment of the microRNA throughout this whole population. There's this thing out here we have some evidence that this one is like associated with microvesicles. So I don't want to give you the idea that gets rid of all the microvesicles and stuff. It knocks it down a good deal, but there's still more for us to do here. The other thing is, notice the relative concentration of these. So here, in the beta purified, according to this, the mRNA is at about nearly 1 mg per mil, whereas in the filtered harvest, it's down to about 20 micrograms. So we get about a 50-fold concentration of the exosomes across this process. Here's flow cytometry before and after. So here's the cell culture that we've been working with. And you can see in the exosome population, there's not really even a well-defined population. It looks like you got just electronic noise in there. You got this big pile of garbage there. You've got material sitting up here. After purification, different story. You can see we've got a really nicely concentrated population of exosomes there. The bigger vesicles are really reduced. These materials are really reduced. Not perfect. There's more to do here, but still, going from here to here, that's not a bad place to start. And just one other comment that I wanted to make there is, we've done this with, serum-free, protein-free, material that's been loaded with BSA and other stuff, platelet lysates, all these kind of things. This method works on everything. We haven't done it on natural samples, so like human-derived or anything like that, biologicals. Should work, but we haven't tried it on that yet. And let's see, we're a little tight. I'm not going to mention the conclusions because they just summarize what I said. However, I will stop with the acknowledgments. We've had some wonderful collaboration with this, and it's really helped us, and lots of folks tested the beta stuff. And if you want copies of this presentation or anything, you can follow up with me at your convenience. Thank you for your attention.
Any questions? So I don't think you mentioned ultra-centrifugation. Is it improving that kind of separation as well?
I didn't mention it because, like in industrial circles, it's something you want to avoid. It's really hard to scale up, expensive to scale up. In the field of virology, also same thing. So once you get towards industrial, work, you want to try to leave that behind, and that's why it's not in there.
So, a quick question on the purification and also the culling. How would we, well, how much information you have on scaling up, and is this only for characterization, or is this really something that can be upscaled to different levels?
This is really eerie because I can't see you.
Yeah.
Yeah, okay.
Yeah, so the question is scale.
Yeah, I got the question. Scale up, you can go as high as you want. So everything you're seeing here was with a 1 mil monolith, and starting off with like 50 to 100 mils of starting material. So we know for like a 1 mil monolith that'll handle probably about 100 mils of feed stream, that kind of thing. But TFF is scalable as far as you want to go. Chromatography is scalable as far as you want to go. The enzyme treatment is scalable as far as you want to go. And one of the feedback responses we got from our collaborators is they liked it because you can go up or you can go down, no problem. So scalability shouldn't be an issue.
Thanks.
We have a question over there.
Here.
Yeah, thanks.
I know you mentioned from the very beginning that you are not talking about biology. I hear that. And you also mentioned that this process, cryptonase and other process, does not damage the exosome.
Mm-hmm.
But have you looked, do you have a plan to look into how removing this chromatin gonna impact the uptake of the exosome, whatever you get, you more pure fraction of the exosome, and the biology associated with. Is this going to alter the downstream, the exo in vivo?
I'll tell you what, if you don't manage chromatin, it will manage you. So as far as the downstream purification, yeah, it massively alters, behavior of anything you want to purify. Doesn't matter if it's antibodies, exosomes, viruses, or whatnot. If you've got chromatin in there, it is interfering massively with your purification. So if you get rid of it up front, you're going to get a really different result than you get without it. However, the result that you're going to get by removing it up front is that you're going to get higher purity, higher recovery, and higher reproducibility of your process. Does that answer your question? And
I was more thinking about the biological property of the purified exosome with your method, because you have not tested that in vivo. So the uptake of this exosome that we get with your method, are they going to be uptaken as similar to what we already know in the field without chromatin removal?
the exosomes should be unaltered. They're not exposed to any extreme environments at any point during this process. And also, back to your comment about, why not heterogeneity? Absolutely, why not heterogeneity? If you look at any one subset of these exosomes, they've only got one kind of small RNA in there, and you have to have a huge assemblage and diversity to really cover, what their overall intended function is. So, the way we do it is, number one, gentle, but number two, it's class specific rather than, enzyme or antigen surface specific kind of thing. So, from what we see and what our intention has been in the way we develop this, you're going to get, exosomes as a class of materials and not, single subpopulations within there. Now, there's one possible exception there is that if you have phosphatidylserine positive exosomes, so for example, from cells that are in an inflammatory state, that has a different charge, and they may behave different, and that's the next phase of what we're doing. I regard this as a foundation. This is our starting point. We expect to make improvements and whatnot, and we'll investigate some of those things.
I agree that it could be, actually, for some applications, the DNA might actually have a functional effect, so, which you might lose.
For sure.
It has
But,
to be tested in every single
but at the same time, that DNA is antigenic, and that's also an issue.
But it can be good for cancer.
Hi there. Can I ask one question? From a GMP perspective, so the DNA, cell-free DNA is something that I think we can probably cope with, but how about the viral contamination? So up to now we're basically dealing with everything being virus-free upstream, but do your separations in any way help with the viral contamination?
Yes. So anion exchange is one of the most common methods for getting rid of viruses and DNA. So that will help there. And I do not agree with you about we can probably tolerate the DNA in industrial settings. I don't think the FDA would just say, oh yeah, that'd be no problem at all. That's something that's going to have to be worked out. So as we're hearing here, there may be beneficial, effects of the DNA or necessary, cofactor types of contributions. But, for just general host DNA and chromatin. The other thing, it's not just DNA. We've all been trained our entire biochemical lives. When you think of DNA, you think of DNA, a double helix on a white background or a black background. DNA is never alone. It starts off with chromatin. It sticks to God knows everything else, in the material. So every time you see DNA as a contaminant in your final prep, that's not just DNA that's pure. That's DNA that's stuck to everything under the sun. And carries big contaminant loads and all kinds of garbage with it. So it's not a casual question. It's going to require a lot of characterization.
Okay, thank you.
Okay. How expensive?
You know what? It's like for my entire career, I've never known what anything costs. It's just like go buy it and stuff like that. So I'm going to leave that question to somebody else who might be able to answer it, but it will not be prohibitive by any means. And, yeah.